Hi, I'm Dan Stanton, the editor and founder of BioProcess Insider, and I am delighted to be joined by Marc Hummersone, the Senior Director of Research and Development at Astrea Bioseparations. Now, Marc joined Astrea in January this year. So, to kick us off, can you tell me about your own background in the bioprocess space and what your role at Astrea actually involves?
Sure, no problem, Dan. So, first of all, starting off at the beginning, I guess good place to start. I'm a qualified chemist, and I've been involved in biotech since finishing my postdoc. And I did that in the total synthesis of natural products or chemistry-related stuff. And I initially applied my skills to drug discovery and development, and I was part of the team that developed the drug called Lynparza, which is on the market now, and that's a PARP inhibitor. And it was about that time that I noticed, an uptick in the use of, or at least people talking about biological therapeutics. And I developed an interest in how they were produced. And it was around about 2006 that it quickly became apparent that there was a clear and unmet need for purification solutions in this new and exciting area of therapy. And a little bit later down the line, shortly afterwards, I had the opportunity to be part of a company who were developing fiber-based chromatography technology for just that purpose. And that opportunity opened up, and I grabbed it, and I quickly became chief scientific officer. And the company was one called Puritify. And Puritify, as I mentioned, was a fiber-based chromatography company. We did well, and we were purchased by General Electric Healthcare for that technology that we developed. And I became part of then Cytiva, as GE became Cytiva. And I was the director of the fiber chromatography department at Cytiva, where I was particularly proud of developing the fiber range of products that are on sale for Cytiva now. And you can click on the link on the internet and you can buy those products. And you mentioned that after that, at the early part of this year in 2021, I transitioned to Astrea, where I'm the senior director of R&D. And what I do here is I oversee the R&D function, and I contribute to the business strategy while developing innovative new products for Astrea and customers alike. And I'm really fortunate to work with a fantastic team who, like me, are dedicated to delivering to the needs of the customer and the patient. And in this regard, I have a particular interest in serving the needs of cell and gene therapy sector. And it's these guys who really require the next generation of purification solutions for their next generation therapies. So that's why I'm here.
Well, we will speak about the cell and gene therapy sector in a while, but let's stick with Astrea at the moment and talk about its history. The company arose out of Pragmatic bioseparations in 2019, and through significant investment, it now forms part of KKR's life sciences, Gamma Biosciences portfolio of enabling technology companies. Can you explain what this means for Astrea and how the company fits in with the other assets within the Gamma portfolio?
Sure, no problem. So first of all, I'm really, really proud to be part of the Gamma portfolio of companies. And Gamma have a vision, and it's to deliver products and services to support the development and manufacturing of advanced biological therapies. And they're committed to advancing the science and the art of bioprocessing. And they want to deliver market-ready innovation and expertise to help those customers who are predominantly in the biopharmaceutical space, and their contract development companies as well, to safely bring their therapies to patients more quickly and efficiently. And in reference to the second part of your question about how Astrea fit into that, I mean, Astrea are a key part of the Gamma vision, and they act at the heart of the innovation engine. As it interacts with all of the other portfolio companies who specialize in both upstream and downstream aspects of biological therapy production. And I'm particularly excited about working with one of the portfolio members, a company called Nanoparal. And they've developed novel fiber-based solutions for the purification of larger modalities. And these could be viruses, plasmid DNA, exosomes, those kind of things, which can't be served adequately by regular resin-based technologies. And this really is where the future of bioseparations is going to be. And Gamma are at the center of it. Gamma are at the centre of it, leveraging on the expertise of Australia. So we're a key part of everything that's going on in the Gamma portfolio.
I think if memory serves me right, you're actually the first acquisition by KKR for the Gamma portfolio. You mentioned nanoparel there. Excuse me if I mispronounced it. What do having these acquisitions and these different elements under the Gamma umbrella mean for your customers?
So for our customers, I mean, we work closely with the other portfolio companies to develop commercialized products using proprietary technology. And so, for instance, if we talk about nanoparel, for instance, we're working very closely with them to develop these commercialized products. They're, for nanoparel, they're highly porous. They've got a fiber matrix. And that allows us to use incredibly high flow rates compared to regular resins. And we're able to impart very high ligand densities on those materials. And that allows the customer to benefit from the very high productivity gains. And this results in, huge time and cost savings. By taking that approach and using the expertise from the different portfolio companies, we're able to leverage on the strengths of those individual parts of the portfolio companies to deliver excellence. And so, again, if I focus on nanoparel and how we're working very closely with those as another portfolio company, our combined goal is to really, really build on all of our strengths, bring it together to deliver high quality products rapidly to the customer who can benefit directly. And so playing to our strengths within all of those portfolio companies is absolutely key.
Marc, this may be a question you can't answer because it was because you've only recently joined the company. But the nanoparel deal and the essential life sciences, the essential life solutions acquisition that Astrea recently made, would those sort of deals have been possible without the backing of KKR and the Gamma portfolio?
I think if you step back and you look at the bigger picture and what Gamma are trying to do with the portfolio, it makes total sense, and the optics are 100% correct. Having the financial firepower of KKR is obviously a driving factor to make that happen. I think it would have only been a matter of time before somebody else had joined the dots in the same way as the Gamma team have. But the Gamma team were there first. They're building on a vision, and it's all coming together. So I think the KKR firepower in the background was an enabling part of the Gamma vision.
And I suppose the following question to that is: are there more acquisitions to come for Astrea with the KKR firepower? And if so, what sort of area are we talking?
So at the moment, we're focusing down on getting our house in order and delivering on all the things we want to within 2021 and beyond. However, we do have we constantly keep our eyes open and ears to the ground, and if there is a suitable fit, then that could well be an option. But right now, I can say that I'm focused on getting things organised within Australia. But like I say, we're always looking.
I mean, Astrea, through various different names and owners, has had a long history. I think it's 30 years within the industry. Currently, you are talking about the discover, develop, deliver approach. What does this mean as far as a business strategy goes, and what does this mean for your customers?
So, the question is, what does discover, develop, deliver actually entail? So, so the discover, develop, deliver is a great way to describe the capabilities of Astrea. And so at the start of the process, you've got discovery, where we're attempting something for the very, very first time. And this often starts with an idea or a concept that's been evaluated and fleshed out in terms of market needs, technical possibilities, problem mapping, and that leads to the developing of an idea. And our fantastic team of highly skilled scientists and engineers take those ideas and develop them and realize them in a quality-driven environment to provide specific solutions through defined processes. And for us, it's not just about the product what's also really important is about how we make the product as well. And then finally, we talk about the delivery part of discover, develop, deliver. And this is about delivery of the product or a solution as a regulated item via our manufacturing arm of the business. And I think that using those words, discover, develop, deliver, captures our capability to provide customers with all of these resources from a single company. And this is really, really important, because if you look out there, there is no other purification solution company with the same in-house capabilities from start to finish. There's no one else out there at all. And it's that ability to have all of the whole discover, develop, deliver under one roof that's allowed us to deliver 21 products that are used in FDA and EMA approved drug manufacturing processes. And we're looking to grow this number. And as such, we are acting as a true partner to our customers in that respect, where everything is under one roof.
Mark, I'm not going to ask you to go through those 21 approved processes, but I'm assuming that most of those are in sort of well-proven modalities such as monoclonal antibodies and such. How are you using this discover, develop, deliver mantra to support the burgeoning cell and gene therapy space?
So it's using that framework that I've just described, and we're applying it. So Australia has the know-how and the capabilities for the discovery, development, and commercialization of advanced therapies. And in order to make sure that we're doing all the right things, we're working closely with industry leaders in the field of cell and gene therapy. And that allows us to gain a deeper understanding of the evolving needs of this rapidly expanding field. And so by understanding the issues being experienced by those working at the cutting edge, we're able to provide purification solutions that are tailored to the accelerating development of these advanced biotherapeutics. And with that in mind, I really did want to mention that we've got, as well as our regular ligand development engine that we have in-house, we've recently supplemented that with an exclusive license to use Avacta Affimer technology for the discovery and development of next generation purification solutions that feature Affimer ligands. And these are particularly useful because, where current technology doesn't work, where we're unable to develop ligands for, specific purposes, we're able to apply Affimer technology to overcome those issues. And, we're working closely with Gamma Biosciences portfolio companies too, and, most notably Nanoparel, to provide not only resin, but fiber-based purification solutions that represent the next generation of purification medium.
That's interesting because, again, you mentioned the Gamma's portfolio of companies which help you provide solutions for your customers. But you also mentioned Avacta there, which unless something changes in the course of this interview, they're not actually part of the Gamma Biosciences portfolio for now. But how important is working with other technology providers in solving some of the issues in emerging technologies? Why can't you do this all in-house, for example?
I think, at this moment in time, we focus down on what we're best at, what we're good at. And if there's an area that we want to increase our expertise, of course, we want to learn in-house, but we find the best people out there doing the things that we want to learn about, and we work with them. And so, developing next generation ligands, we're learning and working with Avacta in order to do that. And those guys are the world's best at developing, these novel ligands. And so it's a fantastic offering under the license agreement that we have that we can provide to customers.
Mark, I must ask you quickly, what are some of the purification solution issues affecting the cell and gene space that you haven't really seen in the more traditional biologics arena?
So within the cell and gene therapy space, a lot of the issues that arise are simply to do with the size of the modalities that we're trying to purify. And in a lot of cases, those modalities are too big to permeate within the pores of a regular chromatography resin. And so, I've mentioned them several times, but working with, if we take our ligand discovery engine that we've got at Australia, and we're able to apply those to Nanoparel materials, which have huge porosities, then we're able to provide solutions that will overcome the issues that resins simply cannot, overcome on their own. There's a place for resins and there's a place for fibers, but we're able to, use the two different forms of matrix in order to provide solutions for just about any purification issue that's out there, regardless, or largely regardless of size of the modality that you're trying to purify. And you can the size of a monoclonal antibody is tiny compared to that of an exosome, for instance, and they require different approaches for their purifications based on the size of the modality.
So I mean, there's no one size fits all solution for purification, or I guess across the industry. But it sounds like you're kind of moving towards a platform that is capable of providing these solutions for, well, any size modality.
Well, you're absolutely right. There is not one size that fits all. However, what we do have within Australia, and as part of the the Gamma portfolio of companies, most notably Nanoparel, we're able to provide a solution for just about all purification needs. And it's that ability to leverage on the capabilities and experience of not only Nanoparel but also Australia that allows us to deliver all of those things to the customer as and when they are required in a bespoke manner.
Okay, Marc, my final question for you is where does Astrea hope to be in 5 years' time, and how do you hope to be part of that vision or goal?
So, if I could rephrase the question, is where we're going to be in 5 years. And, as we are focusing down on the cell and gene therapy market, and the way that we've got ourselves set up, and the size of the company that we are, and how we're able to deliver excellence, using the discover, develop, and deliver sort of mantra that we discussed earlier. We are going to be one of the leading players in providing purification solutions for the next generation cell and gene therapy market. Now, it's one that in many ways is in its as a market, in many ways is in its infancy. However, it's expanding rapidly, and there are many new therapies coming to the fore every single week. We see in journals and so on and so forth that it's expanding so rapidly, and we will be key players in that market to be able to deliver to the biopharmaceutical companies and manufacturers, to give them the capabilities to deliver products more quickly, cheaply, and which directly benefit the patient. So that is where we will be, and we will be synonymous with the space.
Well, I look forward to continually covering every step of that journey. Marc, it's been an absolute pleasure. Thank you so much for speaking to me today and telling me about Astrea and its vision and its history.
My pleasure. Thank you very much for the opportunity, Dan. Really appreciate it.
Thanks. All the best.